Thank you, David, and thank you to the board of ARM and the staff of ARM for putting on another really great meeting here at the Mesa. It's great, the Med, sorry. It's really great to be back together face-to-face in this wonderful location. So I'm going to talk to you today about Agtc, Applied Genetic Technologies. We are an AAV gene therapy company focused in the ophthalmology space. And we are publicly traded, so here's the disclaimer slide for you to read through as quickly as you can. This slide really allows me to provide kind of the 40,000-foot level of who is Agtc, who are we? And first and foremost, on your very far left-hand side, it's to show that we have an asset in late-stage development for X-linked retinitis pigmentosa, it's inherited retinal disease. And I'll talk more about the data that we've already released for XLRP and our timing for releasing data on the 2 registration-facing trials that we have in progress. In addition to the program in XLRP, we have 2 additional clinical programs. One of them is a chromatopsia, which is another inherited retinal disease, and we are expecting feedback from the FDA this quarter on our end of phase 2 submission so that we'll be then able to guide everyone on what the late-stage trials for a chromatopsia will look like. And then our final clinical program is in partnership with Bionic Sight, a medical device company in New York City, and it's in the optogenetics space, and I won't have time today to really talk to you in great detail about this program, but it does have a very unique technology. We're using a light-sensitive protein developed by Ed Boyden at MIT that has a much larger range of activation than some of the other light-sensitive proteins being used in the space. And also, it's a very unique medical device. It is a wearable pair of goggles, but it doesn't just blast light. It doesn't just enhance the light. What it does is it uses an algorithm to repulse the light so that the retinal ganglion cells that are being transduced can better understand the light waves and the patient can experience finer visual acuity. So we're very excited about that program. The company is really built, and what we're really known for is our integrated gene therapy platform. We've been in this space a while. We were founded by 5 academic scientists who had both medical backgrounds as well as PhD and virology backgrounds. And we have developed for each of our programs a very broad set of preclinical data that has told us how to get into the clinic with the highest level of potential success. And I'll talk about that more in a little bit. And then manufacturing. There's been a lot of discussion about manufacturing at this conference. And we have developed our own proprietary manufacturing method. There's very few people that use the method that we have. And from just 40 liters in suspension, we manufacture 2,000 doses for our ophthalmology program. So it's a very efficient, effective manufacturing system. And as you'll see later on, we just made a big investment in this system to bring our manufacturing internal to the company. So this is our pipeline. And as you can see at the top of the pipeline are those 2 later stage programs, XLRP, which I talked a little bit about, as well as a chromatopsia. XLRP, we are set up for a gangbuster set of data releases over the next 2 to 18 months. And there's 4 sets of data coming out. The first is 3-month data from our Skyline Trial, which is the first of those 2 later stage trials I talked to you about. The second is 24-month data, so some durability data from our phase 1/2 trial. And then there'll be 12-month data. This is a typo. I haven't learned how to travel in time yet. So it'll be 12-month data from the Skyline Trial in the first half of 23. And then the interim data readout on our second late-stage trial in XLRP, also in the first half of 23. So 4 separate data readouts over the short term. And then, as I mentioned, we'll be getting that end of phase 2 feedback from the FDA this quarter, and then be able to talk about what that late-stage trial will look like. For the optogenetics program, the next step is the high dosing. They're doing the high-dose cohorts in that trial, and we'll be releasing then the high-dose data from the RP program. And then the rest of our pipeline is preclinical, and I don't have time to spend much about it, but you can see we're diversifying into hearing loss, we're diversifying into CNS indications, and really my team has done such an exceptional job over the last couple of years. They were able to secure all the non-human primates to conduct this year 3 different toxicology and biodistribution studies, which means we may be in a position in 2023 to move an additional 3 programs into the clinic. So we've really been successful at managing that supply chain issue and really networking our way to be able to still move our programs forward. So X-linked retinitis pigmentosa. So this is our lead indication, and I wanted to tell you a little bit about this disease. It's a fairly large indication for an IRD, about 20,000 patients in the US and the EU, and there are no current treatments for this disease. And it's a very devastating diagnosis. These kids are diagnosed between the ages of 6 and 8. At first, they're labeled clumsy because they start running into things, because the first thing that goes is their peripheral vision. And finally, their parents become concerned enough that they get to the doctor, they end up at a retinal specialist, and then they're told that they're going to be blind, legally blind by 45, and probably completely blind in their 60s. So you can imagine what a devastating diagnosis this is. The parents, there's nothing for them to do. The kid just needs to learn how to react. And he probably knows a relative, an uncle, a father, a cousin that has this disease and has seen them lose vision over time. So it's really impactful to these patients. So this is the slide that I try to use to give you an idea of what's a little bit different about what we do at Agtc. So we really know, and we call ourselves data geeks because we really love the science, we love the technology, and we also understand that once you get that clinical data and you're preparing your package for the FDA or the EMA, it's not just the clinical data you have to show them, it's all of your preclinical data, your safety data in animal studies, your proof of concept studies, your potentially long-term durability studies in these animal models. And so we have spent a lot of time right from the beginning making sure we were developing all this data, and we screen very carefully each component of our vector in an appropriate animal model. We don't skip the non-human primates because we know tropism isn't maintained across species. We don't skip testing whether the promoter acts the same in a primate eye as it does in other eyes. And we also use naturally occurring animal models, large animal models. For XLRP, there were 2 different breeds of dog that have the same genetic mutation, and we did extensive dose ranging in those dogs. We looked at the histology, made sure there was expression, made sure that there was actual function going on, made sure those photoreceptors really were maintaining their health over time. And we think the time we spent here, which our competitors did not, has allowed us to get the robust clinical data that we have today. And we think this is a very important and differentiating component of the company. And this is our phase 1/2 data. It goes out to 12 months, and as I said earlier, we'll have 24-month data late this summer or early fall. And first and foremost, the product was generally safe and well tolerated. We did not see any dose-limiting toxicity. We did not see any late-stage inflammation. Our inflammation was well controlled with the steroid regimen we have. And we think the broad safety profile we saw over an 80-fold dose range was because of our manufacturing process, our careful preclinical work, our screening, our surgical training, and all those things together meant that we could provide for the patients a very safe product, and we think that's job one. Job one is safety, patient safety. But what we also saw in this phase 1/2 trial, in somewhat of a dose response kind of way, is especially at the higher doses, we saw multiple signs that our product was having a positive effect on these patients. First, we saw improvements in visual sensitivity in greater than 50% of the patients treated at the high dose. Second, we saw trends in visual acuity. It's very hard in this patient type to see true three-line improvements in visual acuity, but the trends were very supportive, as you can see from this graph. And third, we saw improvements in the actual structure of the retina. And so even though in orphan development and in these kinds of trials it is a small n, when you start to see multiple endpoints line up and correlate to each other, that's when you really start to believe there's something going on here. And this is what led us to our end of phase 2 discussion with the FDA and into late stage development. So the 2 trials that we're doing in late stage development, the first is the Skyline Trial, and that's the data we're going to be reading out this quarter. This was an expedited path for us to get data really quickly that in a separate set of patients that were randomized and blinded into 2 different dose groups that we could show we could match the data in our phase 1/2 trial. The other thing we did is we added a new endpoint. We added in a mobile navigation challenge, otherwise known as the maze. And this is a maze that has been well used. It's very similar to the maze used to approve Luxturna. And so it would give us an opportunity to help people translate the quantitative numbers around visual sensitivity to some real-world evidence of patients really doing better in their daily living and make their way through the maze. And what's important about this maze is that it is done multiple times, and in between each time the patient does it, the maze is changed. So there's no ability for the patient to memorize or to just know where they're going. It's a very complex maze, different colors, high obstacles, low obstacles, patterns on the floor, and all of that is changed each time the patient takes the test. So we're very excited to be able to see this data this quarter to see how that maze helps us understand the breadth of improvement we're seeing in the patients. The second trial we're doing is the Vista Trial. This is a trial that was negotiated heavily with the FDA based on their input. They thought it was very important because of some of the biases that can be introduced in ophthalmology endpoints to have a totally untreated control arm and then also blind in the low dose and the high dose and compare those 2 dose groups to the untreated control eyes, not to the fellow eye. There's no way you can blind the patient to which eye was treated. They know that. And so it's better to compare to the untreated. So this is a statistically powered trial. It's based on visual sensitivity as the endpoint with also supportive endpoints of the maze again, supportive endpoints of visual acuity. So we also have patient-reported outcomes. So this will be the second of the 2 trials we're doing with the early data coming out in the first half of 23. Our second program is a chromatopsia, another IRD, fairly large patient population, about 14,000 patients, very different indications in that it's just the cones that are affected. These patients are born blind, they're born sensitive to light. They would be in excruciating pain right now with these lights on them. They would not be able to tolerate it. And they also only see in black and white and shades of gray. And we've also done a very similar trial in a chromatopsia that we did in XLRP, dose ranging over an 80-fold dose range. And once again, we saw a very good safety profile, and we saw significant improvements in visual sensitivity. And these improvements translate into a 16 to 24-fold improvement in visual sensitivity in these patients. And importantly, it also lessened their light discomfort, so they would be more comfortable with these lights on them. So the news about manufacturing is that we are actually bringing it in-house. This is a photograph of where we are with the construction. It's a 20,000 square foot facility, and we're taking over the keys in September and doing all our shakeout runs in the fall, and we'll be doing GMP manufacturing starting in 2023. Finally, just in the last few minutes I have, over the last 9 plus months, we have grown our senior management team. We are planning for success. We're planning that XLRP is going to be approved and we're going to be commercializing it. So we have put together the team that really understands ophthalmology, really understands gene therapy, really understands commercialization, really understands finance and raising money, and in this environment, we all could care about that. And I'm just really proud of the people we've been able to attract to the company and the experience that we have in order to propel us forward for success. So thank you for your time. I hope to talk to many of you later today.